So like here's the recognition sites. As I told you, what's the work of restriction? To cut from the mid, like from mid of the DNA, like in gene of interest, they will cut mid, and then exonucleus it was cutting from the ending. So now what we'll study here? Here's the one term which is known as, wait, let me set it's here. Okay, so now that term is palindrome. So now what this is, like this is the like name of genes, even we can say name of DNA, we can say, like by if we are reading here, like if we'll read from this end, the meaning would be same. If we will read from this, the meaning would be same. Like from wherever we want to read it, that would be same. So the example can be Malium. So like we can say Malay. This means mal, like Mala. Y is in mid. So like this name is known as palindrome. Okay, so like this type of like known as palindrome. So now if we move towards the example, so as we know there's the 5 complement, here's the 3 complement. So if there's 3, so this means the opposite, it would be 5 here, and here would be 3. So now if you wanna like add nucleus, like this nucleus not, if you wanna add the proteins, so here we are adding, first one is CC, second one G. So here as I told you it would be reversed. So here is the G in the end, so now here would be in the starting G. Here's the C. So now what will happen here, as I told you, we can read it like this. So now this is the example 1. Now if you move to another example, so here's the another example. Again 5 complement, 3 complement, here would be opposite, 5 complement, 3 complement. So if you wanna make another example, so this is the GAATTC. So if you wanna do opposite of this, so it would be started from C, here's the T, second one, after that T here, then A, then this G. So now what will happen, this was the step 2. Like here we have taken another example, I was just showing you the like this term. Now we have taken example to prove or like to know how it's happening. So this is the example. Now if you move, if we want to write it here again, like here's the same as I told you, 5 complement 3, here would be opposite 3, here would be 5. So now as you know, like these had gone divided, and the bond between them, that bond is known as hydrogen bond, and this one, like this line, is known as phosphate. Okay, so now here what will happen here, as like we wanna cut this, as I told you, like this recognition site or restriction can cut from anywhere. So this is the first, it is cutting from here, and in this one it is cutting from this one side. Okay, so now when these would be cut from here and here, so we're gonna make it separate structure. So let leave here, there's no any more space. So if I wanna make here, here's the first one G, like as it got separated, and still this having bond with this one G, and it's bond having bond with the C, the same CCTA. Okay, till this. And if you wanna make another, so this is CCTTA, like this the one structure. Now this one got separated, so what will happen here, here would be G, this would be having bond AATTC. Okay, so this was the like another, here we can say this got separated, like this the same we have just written in the separate form. So now we want to see, like this mean like these all are still unpaired, and here these all are unpaired. Okay, unpaired. So now what will happen, like this ends, now still like this ends, like these all are still sticky, or like they are having sticky ends. So this means still they can make a bond with another. So now this is also known as biotechnology, like here they can add with another to make another thing, this would be add with another to make another thing, like guys, like the biotechnology is used like this. Okay, so this was another example. Now if we move towards the, like here are the terms. So the term which was palindrome, which means like from where we want to read, or like they would be having same meaning. The Maliums, this was the term. Here's the example as I told you. So now if you move to furthermore, so as I told you, this is the cell, like this is the bacteria cell, and they are having, as I told you, main DNA. In last video I have discussed about that main DNA, and inside that there are the plasmid. So now like these plasmid can, like here's the plasmid, the round like structure is known as round like structure, or even we can say they are like double structure. Okay, so now what will this do? It will transfer gene, like transfer gene from like one to another. Like if this wanna move from this bacteria to another, it can easily move. Likewise, this is the work of the plasma, like transfer gene from one to another. Okay, so now this was the recognition sites. Thank you.